Mark Centillion. I'm in the Department of OBGYN. And the reason why the smarter Dr. Centillion is not speaking is that she's on an NIH study section right next to me. This all came from so Donna and I came here in 2006, and we needed tissue for our own studies. And so this has really kind of blossomed since we came here. I am the clinical research director for the Maternal Fetal Tissue Bank, and Donna is the PI of the Women's Health Tissue Repository, which is a much bigger bank that encompasses our entire department. So we'll get into that in a little bit. So as the Women's Health Tissue Repository, or WEATHER, as we like to call it, is really a repository of 5 distinct biobanks. The Maternal Fetal Tissue Bank, PATCH, which is a related bank, which is Paternal Contributions to Children's Health, so we can get genetic triads and sample triads, as well as clinical data triads. Well Women Bank is a sort of a catch-all biobank for those who are healthy, otherwise getting well women care. Most recently, our urogynecologists are using this for kind of uterine prolapse care and kind of urology issues in older women. There's, of course, the Reproductive Endocrinology and Infertility Bank, where we grab a lot of tissues, not just the usual blood samples and plasma and live cells, but also fluid and media that come from IVF cycles. And then, of course, the Gynecologic Oncology Bank, which is just a huge bank of tumors and clinical data. Really, the Gynecologic Oncology Bank has been going on in our department for 30+ years. When we came in 2006, we started to kind of coagulate everything into a sort of a singular force. And so the reason why we have these different biobanks in these different areas is that you don't want to give a pregnant mom a consent form that says, "Hey, you have cancer. We want a chunk of your tissue." So it sort of allows for a good each of these groups and each of these banks have their own clinical research director, but they all fall under Donna's auspice as the PI of the entire repository. So why this? Why a women's health tissue repository? Mainly because women were sort of left out of everything. In 1993, the NIH Revitalizations Act, it surprises me it took that long, where they actually required women in studies. And it was even blew my mind that it was only until 2014 that NIH began requiring applicants to address sex as a biological variable. And I know that everyone's doing that on their grants. And so, even though we have lots of data on men and women, and even less so in pregnant women, rural pregnant women is sort of Iowa's bag. That's sort of our gig. We have sort of a very unique set of exposures that could affect these women, and we're able to provide very person-level data to address many different questions. So this is from our paper that we wrote in 2014, just to show the overall structure of the umbrella bank and all the sub banks underneath it. Our inclusion criteria overall are greater than 18 years old, pregnant, non-prisoner, HIV and hepatitis negative. For that, because we don't, this is basically run out of our lab here. So we don't have the facilities to deal with higher level infections, English speaking, and mentally competent. So you may be wondering what this picture is. This is an Italian sculptor who makes these kind of sculptures of people coming out of walls. And this is my reminder to tell you that we are effectively embedded into the clinical workflow of all of our banking. So for example, the Maternal Fetal Tissue Bank, and that's part of the reason why we have very successful enrollment. We have over 8,000 unique MRNs, so that's about 4,500, over 4,000 mom and baby dyads. We have the REI Bank, the Infertility Bank. That's about 1,000 women and partners. While Women Bank is still building. PATCH is something that we've put in as people want more triads. The Gyn Onc Bank, like I said, has been going for a long time, but they have about 600 or samples. And then even beyond, even though a lot of the samples and the clinical data that we are collecting originate here from the University of Iowa, because of the work we've done from the bank and that has fed other studies, I've established other kind of outside networks outside of our, hollowed halls here at UIHC. So I have a network that was AHA funded, where we collect samples and data from a lot of outside sites, Waterloo, Des Moines, West Des Moines. So it's really sort of this kind of regional bank. And in fact, we do have access to more international samples with our work with a international perinatal biobank group, where we share data and samples. But so our reach is actually a lot further than what we're just seeing here at UIHC. This is just an example of our average enrollment. This is actually a little lower. Maybe this is because of COVID, but overall we get about 63 moms consented per month in our maternal fetal tissue bank. And part of the power of it all is that women and participants don't experience anything different than their clinical care, because we approach them as soon as they begin clinical care, whether it's in the OB unit or the gynecologic oncology unit or the REI unit. And anytime that they have routine blood draws or a procedure, we are IRB approved to grab a little bit of what was grabbed. And what I mean by that is that it's not leftover samples. It is a separate tube. It is a separate collection of placenta. It's a separate collection of cord blood, so that they can be used specifically for research only. And that way we can have a really good QC on the quality of the DNA, RNA, and everything else that we get from those samples. So for example, the maternal fetal tissue bank, we get everything: whole blood, urine, placenta, cord blood from the kids, and all the blood gets processed into plasma, into viable mononuclear cells, and we freeze them down for viability. So a lot of my work in immunology and pregnancy relies on those kind of cells that we can unfreeze and actually do something with in terms of immunologic outcomes. Patch is all whole blood, while woman is actually whole blood, and we have some surveys that are embedded into the bank, as we do now with MFTB, but they're also collecting a lot of urine. What's unique about the REI infertility bank is that we get follicular fluid from first and pooled from IVF cycles, as well as seminal fluid, and of course gynecologic oncology has tumors frozen, as well as DNA, RNA samples, as well as whole blood. So sample processing is done the same. This takes a team. We really do have a village here that helps us go through all of these banks. We pick up samples 3 times a day from our phlebotomy lab, L&D, and clinics. And when I say we're really kind of metastasized into the clinical workflow, we really are. So for example, somebody comes into labor and goes to labor and delivery. The nurse gets a flag that says, hey, this person's an MFTB. Well, they will know to draw, in addition, right before they put their IV in, they know to draw our additional research tube for blood, and then they know to flag us and flag the residents to collect our placenta and to collect the cord blood, and that's a delivery sample. Same thing happens in the outpatient center, where they know someone's at MFTB, they get flagged. The phlebotomy lab, because they're not doing anything extra, it's not an extra poke, they allow us to throw on an extra tube of blood on their collection. So all of these are designated research-only tubes. We use an ACDA tube, which is a citrated tube. We specifically chose this tube because it gave us the best return with regard to plasma, as well as better return on having live cells to do kind of immunologic experiments on. And further, because it's not magnesium as you would in EDTA, it doesn't affect any other sort of assays that you may be dealing with. So they get processed in the plasma and peripheral mononuclear cells. They get 4 plasma vials in our -80s, and then the live cells get stored in liquid nitrogen. This is all very protocolized. We have 12 -80 freezers, and we're planning to get more. 8 static liquid nitrogen tanks. Everything is on a temp track system. So my phone, Donna's phone, our pagers, our entire there's a hierarchy of people where our devices explode whenever there is a freezer going down below specs. That happened to us 2 years ago on New Year's when a pipe broke and it started flooding our lab, and the first reason why we knew is because temp track was going on saying, uh-oh, something's malfunctioning. So it has been actually helped really save our lab. Queena mentioned this before. We are involved in Lab Matrix, or UI BioShare. The UI BioShare is a conglomeration of all the different banks at UIHC. And Donna was on the kind of the committee to help harmonize not only the way we collect samples, but the way we collect preliminary data. So Lab Matrix is actually a web-based lab information management system that the college and the Cancer Center bought for a cool price of $1 million. And so what this does, it's great because it's a very simple query interface where you can take chunks of data, so your subjects, your pathology, your biomaterials, and you can easily put together a query so you could identify where your samples are according to what they have. So Queen had mentioned it before. So for example, my lab is very interested in vasopressin and osmolality and all this kind of stuff, this kidney sort of stuff. And so we put that data into Lab Matrix. But what's also uploaded into Lab Matrix is a lot of basic clinical stuff that's important. In my lab, whether or not you're preeclamptic is very important. So that stuff is already there. So if I want to run a query looking for all 18-year-olds with a first trimester sample, first trimester plasma, and has a copeptin, but I want to write in the osmolality, I can put that query in a very simple kind of, as what you're seeing here on the screen, a very visual way, and it'll tell you, Mark, go to freezer 5, box 6, slot 2, and they'll give you this whole list. So you can go literally from clinical data to samples in one fatal swoop. One of the other things that Donna and I have worked on since we got here, so that's what I talked to you about, is sort of the architecture of the maternal fetal tissue bank and our biobanking efforts. So that's how some of the clinical data in the bank is linked to our biosamples and how it's organized. Now, we are increasingly frustrated that overall, and I say this as a clinician, as an MD, that doctors suck in documentation, right? And so at the end of the day, the data are reliant on what's going on in what's written in the literature, in the medical record. So this is literally what I'm talking about now. This data mart has been a labor of love for Donna and myself for the past 10 years. We, along with our friends in the biomedical informatics group in the ICTS, have worked on a brand new data mart structure. And so what this structure allows us to do is to take multiple sources, so Epic, other EMRs, or our data, and consolidate them into a singular data mart and have a common DIA, or what we call OBDIA because it's obstetrics, data integration structure, so that everything is solidified, and so you don't have to look in an EMR to grab that data. When I talk to people about this, I often say this is sort of like the Hoover maneuver for clinical data. We can literally suck data out of our electronic medical records, and I'll give you an example of that. And so by being able to integrate these multiple sources, now the reason why it took 10 years is because, like I said, doctors suck at in documentation, and so part of that, and Marco knows this too, for example, one of the distinct data elements that we were trying to clarify is the gestational age of delivery. It would make you would think it'd be really easy to find in the medical record. When we looked for that data on the back end, on the relational database that feeds Epic, we found 6 answers. So we spent a lot of time. It took us 2 years to get to the variable that where now if I say gestational age of delivery, I can suck it out of Epic and I can be sure that that's the case because of years of beta testing. So a lot of this goes because we have a common language between, and so we're able to do this with multiple sources, but even different EMRs. And we're starting this venture right now, actually, with one of our outside sites in Des Moines, where we're going to try to kind of get these variables to align so you have a standard vocabulary and get data quickly. So why is that important? Well, crap in, crap out is the truth of clinical data. This is an example of what this Data Mart viewer can do. So this is kind of limited access, only Donna and I have access to this viewer at this point. But you could see there's a whole bunch of saved queries. So I talk like I know something about computer science. The answer is I don't know squat about it. I know in general how databases work, but I am not somebody who works in SQL. I loathe SAS, even though I know how to code in it. I hate it. There's simpler ones I like to use. So they made this Data Mart viewer so that someone as stupid as me can easily get to the data very quickly. And so I'll give you an example. So these are kind of basic queries that are already put as part of our viewer. And because you guys are not IRB approved, I got rid of all of the HIPAA identifiers. But I click on mom episode. So in our EMR, episode is kind of like something that happened. So like a pregnancy episode. Each pregnancy episode has a pregnancy episode ID that links mommy data and baby data. So what we have access to is all of the pregnancies and all of the episodes that have been associated with Epic since the dawn of Epic, so around 2009. So as you can see, in terms of mom episodes, we have about 61,000. And so there's another query called mom diagnosis ICD-10. So you could pop in an ICD-10 code and you could find out of the entries of the 2.2 million entries of diagnoses in this Data Mart, I can find whatever diagnosis I want. And of course, since I am myopic about preeclampsia, 014 is the ICD-10 code for preeclampsia. I put it in this little box. It's completely web-based. It takes 7.42 seconds to run, and it comes back with 5,000 plus entries. So this is a very quick way for us to kind of see, all right, who's got preeclampsia in the whole enterprise of that is documented in Epic. And so this has been a very powerful tool for a lot of studies that not only we're doing, but studies that our collaborators use. And so I'll talk a little bit about that. The key to this is this pregnancy episode ID. That pregnancy episode ID is something that you'll never be able to find on Epic or the EMR. It's a Epic generated tool that links mommy data and baby data. And because we have that, we can we have access to all the labs, all the procedures, all the medications, and we can have it spit out in seconds. And so this has been a very powerful tool. And the one thing I didn't mention too, that we are not a core. We are not charging anyone to work with us. But how we charge you is it's a collaboration. And everything that we do is a collaboration. We do it like Donna and I often say we're the best biosample and clinical data drug dealers, because we'll give you what you need to begin with, but then let's build an empire together, right? Let's build a new study together. And that's literally for collaboration. We were not out there to build this huge resource such that, we'll hog it for ourselves and get ourselves paid off of it. It is literally a collaborative effort. So we have international, national collaborators that are using our resources here. So how do you think this can accelerate the research? So when we're talking about whether if Marco comes to me and says, hey, Mark, I'm interested in first trimester samples, I say, okay, we'll have you talk to Donna. You need to fill out a user agreement. This user agreement basically just says you won't de-identify it, because the magic of what we do is that it is in the bank, it is not considered human subjects research, because we, as the honest brokers, will scrub all of the HIPAA identifiers out of your data set and out of your biosamples. So you fill out this user agreement that says you won't share it with anybody else, and you won't de-identify the patients. You fill out a human subjects determination form from the IRB. They say, yep, you're using the bank, and then you get a IRB number. So sometimes it helps when you're applying for grants and when you're writing it down on papers. And then you get a whole bunch of fun, coded, de-identified clinical data and biosamples. The data mart's a little different. The data mart, of course, remember, it's literally, think of everything that's sitting in Epic. That's our data mart. It's not a static database. It is you go to the data mart, shop for what you want, and you pull it out, and it gets dumped out. There's an umbrella IRB for obstetric studies, and then you get clinical data. This is on REDCap, and I'll show you how to get to that. It's pretty much email Donna, but what happens is, if you want to get identified data, you can, but you have to go through this process. We add you on as a modification to this current retrospective obstetric research study. So how this accelerates research is that in these two resources, you don't need your own IRB. You just have to have the idea, and we can help you through it. So let me give you a few examples of how this has actually worked in my lab and with Hans. So a couple of years ago, I had an idea with a friend about measuring vasopressin in women as a predictor of preeclampsia. So we showed that our preliminary data that we got showed that this marker of vasopressin was highly predictive, with ROCs in the 0.9s of predicting preeclampsia as early as the 6th week of gestation, as soon as sperm and egg meet. So that exciting preliminary data got us our first AHA grant as an innovative project grant. It got us a patent and a paper that was the basis of a lot of the work that my lab does right now. So this paper is not just a clinical study showing that we have a brand new biomarker in the development of preeclampsia, but we used that to translate it into a novel mouse model that also recapitulates everything we know about preeclampsia. We applied for this large and received this American Heart Association $3.7 million program grant, and that has been also the kind of the push to get further NIH funding, my R01, some CTSA funding, another AHA grant that looks at postpartum samples, the building of our national and international cohort, and a bunch of publications. The reason why this wasn't a Mark Pettin himself on the bat, but what the important point of this is, from this point to the point that we actually put this application in was less than 18 months. And so this really kind of pushed our work very quickly, because we could go, hey, I got an idea. We went to the freezer and we had everything done. One last example is what we're doing now with Hans and crew. So Hans, Weibao, Darren, Dave, me, and Donna, we sort of have this environmental perinatal research group. We have all this data, and there's a lot of associations with perinatal disease and environmental exposures. So let's get all the people in the room to talk about it. So we've been meeting for, I don't remember, Hans, it's been a year now or half a year? I don't remember. Something like that. And so we did that because we know that environmental exposures affects everything, particularly in pregnancy. Not only does it affect mom, but it affects baby, and then that baby's baby, right? So maternal exposures in pregnancy are crucially important to future health, not just to women. So of course, my drug of choice is preeclampsia. So we're trying to ask this, just had this broad question of looking at associations of environmental exposures and preeclampsia. So we started off with our cohort in the maternal fetal tissue bank, and we kind of geolocated them in terms of where are my preeclamptic. So this is the smaller cohort of MFTB of about 4,500 women. So we start seeing these clusters of preeclampsia, and we're trying to figure out why. One of the thoughts was, is there something in the water? And so Dave and Darren and Hans have been kind of looking at sort of higher level state data with regard to what's going on in Iowa in this group of women. So what they and so they looked at the overall sodium levels in that community's water supply. What we were able to see is that less than 20 milligrams per mil was sort of like the baseline, because that's like I think the maximum that you can have according to law in your water. It has to be less than that. So and then we looked at odds ratios for preeclampsia. So this is over 31 communities, and we see this clear kind of dose response relationship that in communities with higher levels of sodium, you have a higher degree of preeclampsia. And so Darren did some calculation of saying if women drank 8-ounce glasses of water, women can get their 50% of their RDS of sodium from water alone, never mind their diet. So this is very exciting from a mechanistic standpoint. This is brilliant because in my mind, my work in immunology and the vasopressin and the problems in the kidney is completely associated with sodium handling. So what we're doing now is going to the data mart, looking for, instead of just looking at that 4,500, we're going to look at all of the pregnancies to try to look for this relationship. And then we have the opportunity to go back to the bank, grab some of those biosamples, and we can actually dig to your question before, Diane, to dig into some of these sodium-related pathways and vasopressin pathways that my lab is particularly interested in. So this has been a great way to, and that, again, we haven't been meeting for that long. So how do you get involved? Email Donna. She is sort of she is the center of it all, and we will be more than willing to discuss collaborations. We certainly have a clinical research kind of crew here, and we could help you with IRBs, talk about sample availability and resources. And the one thing I do want to thank is everyone in the lab. This literally takes a village. We have a bunch of people in the lab that do everything from picking up samples to consenting to processing. So this is all a big, huge effort by a lot of people. So it's not just me and Dee. So with that, I'll shut up.